Now, when we remove the estrogen receptor from these neurons, that basically that makes these neurons blind to estrogen, and that basically, from the neuron's perspective, dramatically increases the progesterone to estrogen
Right.
ratio because
Right.
now they only sense progesterone but not estrogen.
Mhm.
And then what we see is that we see a dramatic increase in the observed probability with which these animals become aggressive. So it really scales with this with this expected relationship between the ratio and the aggression probability. And when we did the opposite, so when we removed the progesterone receptor, now the progesterone to estradiol ratio, as experienced by these neurons, is actually much lower. And hence, the probability with which they show aggression is also much lower. And that really told us that there's, in fact, it is the ratio rather than the individual levels of these hormones that are being sensed by these neurons.
Right. And remind me, which neurons are we talking about? Are these the medial preoptic area neurons?
Exactly.
Got
Yeah.
it. So the hunger neurons are in the arcuate nucleus. They're projecting to this part of the hypothalamus. This part of the
Mhm.
hypothalamus has the estrogen and progesterone receptors. And so these neurons in this part of the hypothalamus are integrating a sex hormone signal that's telling you where you are in the estrous cycle, together with
Mhm.
a hunger signal that's telling you how hungry you are.
Exactly. That's how it seems to work. Exactly. Yeah. And then
Interesting.
and then the question is basically, okay, how do those completely different signals, how are they actually integrated? What
Right.
happens inside those cells? And in order to find out what's happening there, how these neurons are capable of integrating those two very different signals, we turn to a technique called patch clamp electrophysiology. So that's basically, a technique where we take acute brain slices from a mouse brain and then culture it in a nutrient-rich medium for a few hours, and then perform electrical recordings from individual neurons. And they are what these recordings are extremely sensitive. They can really detect very small biophysical changes in these neurons, electrical changes, but they can also allow us to then afterwards reconstruct the morphology of these neurons. So it's a very labor-intensive but powerful and very sensitive technique. And what we found was that both of these different signals, so the hunger signal and the hormonal signal, they converge on a single ion channel. So basically a pore in the neuron membrane that regulates ion flow into the cell and regulates the cell's excitability. And both of these factors, these signals, seem to be converging onto one such type of ion channel, the so-called HCN channels.
I see. So putting this stuff together, basically what it sounds like is throughout the estrous cycle, hormone levels are fluctuating, the relative ratios are changing. In
Mhm.
particular, a key ratio here is the estradiol or estrogen to progesterone ratio. As these two things change, right, one goes up, one goes down, the ratio is changing, these hormones are getting into the brain and the
Mhm.
ratio is changing, and both of them can get into these neurons, and the relative ratio will dictate how much of this ion channel gets made in these particular neurons. And as a result of how much of that channel there is, it changes how excitable or sensitive to its inputs the neuron is, and therefore how much it can listen to, like the hunger neurons and other inputs it might have.
Exactly, exactly.
I see.
That's it. That's the one layer of regulation. Exactly. That's how these hormones affect the channel expression. So via the basically the gene expression level, and then how hunger affects this ion channel. That's via this neuropeptide NPY that these neurons release, and this neuropeptide binds to certain types of neuropeptide Y receptors, and then there's an intracellular cascade that we don't fully understand yet. That's downstream of this receptor binding event that communicates directly with these ion channels and also downregulates the function of this channel.